Thank you very much for your kind introduction. Yeah, I've enjoyed my 2 visits to Costa Rica tremendously, especially the visit to the coast, and then also in Bio, and then at the university the last time was also great fun to come visit and teach another course. And of course, hi Godofredo, I think you're there. I saw your name on the list, so good to see you again, or good to, well, you know, be there. So yeah, as Giselle mentioned, I would like to talk about some of the work that came out of the European project, but some of it is really just the general interest in extremophile natural products. I'll explain where we are. So we are in the north of Scotland, so in this little star here. University is is more than 500 years old, and founded by this bishop in 1495. And this chapel was built first of all. I finished in 1514, I think, and very beautiful. I walk past it every day to go to work, which is very nice. And it's still a great university to work for, so it's very nice to be there. Over the years, we started off when Godofredo was there. We still had a very small laboratory and some space in the main lab. But then in 2010, we're able to found a new research center, and that is really the aim is essentially this: is to find new molecules from nature to treat various different diseases, and that would be things like cancer, infection, inflammation, parasitic diseases, epilepsy, and Alzheimer's disease in particular. And quite often we're interested in the functions as well. So we're doing some work right now on sea slugs called nudibranchs and their interaction with the environment and with their food sources. So it's an interesting project that's just beginning on chemical ecology. The tools that we use are the classic tools of chromatography, spectroscopy, and now also molecular biology helps us a lot. And we do some synthesis, molecular modeling, and we have been involved in software development, although that's less happening less right now. The people involved are myself, Rana Abel, who also worked with Phil Cruz in Santa Cruz, the guy that I wrote the textbook with. Dr. Deng has been with us for 12 years now and is an expert in particular types of enzymes, in particular those that do chlorination and fluorination. Dr. Hussein is a research fellow that has got his own independent research program now in synthetic biology, looking at the kind of pathways I'll describe later. Dr. Tremblot is an organic synthetic chemist. He's very useful to have on the team to synthesize substrates that we feed to biosynthetic pathways, and you can see examples of that later. And Russell Gray is our NMR technician. We just lost our mass spectrometrist, which is a bit sad. She went off to Austria. The lab itself is about 300 square meters. It is a chemistry lab for 10 people, a drying room where the noisy things are located, like the freeze dryer, a micro and molecular biology lab, cold room, a clean lab for the analysts that work in the same space as us, a spectroscopy lab containing 600 megahertz and a 400 megahertz NMR, and 2 mass spectrometers, a Bruker Maxis, and also a Thermo Orbitrap. And again, everything is, well, the 2 mass spectrometers are broken right now because of the difficulty getting an engineer in a lockdown situation. There's a space here that's taken by a spin-out company. And we have, in addition to this, we have another molecular biology lab over at the medical school that Dr. Hussein has taken. And we have another microbiology lab in a remote location to keep long-term cultures going. The group is very international. It always has been. It's very nice to work with so many people from all over the world. And part of that is for us to have good interactions with countries where exciting biodiversity exists and collaborate with them. And secondly, it's just a really, it makes a very vibrant atmosphere to have students from so many parts of the world working together. So why are we interested in natural products? I don't think I need to convince you, but I just want to reiterate many of the things that we say to each other when we're talking about natural products. And first of all, it's the beautiful biological diversity that we have. So these are microbes from the deep sea. And then the genetic diversity, so that not only are they biologically diverse, but they show amongst these species that we discover a huge range of genetic diversity between them. So we can have organisms that are very distantly related that we investigate. And of course, each one of these organisms, when you look at their genome, may have a huge biosynthetic capacity. And we're looking at things here. This is the Solanospora strain, just as an example, dedicates 12% of its genome to biosynthesis of small molecules or specialized metabolites. And I think it's 30-something clusters, of which we've perhaps seen 10. So again, there's a lot of interesting things that we can look at. And of course, that then generates really interesting chemical diversity. And most of those molecules will have some function in nature. We don't know what it is quite often, but we might detect that these things have new mechanisms of action. And I've got a couple of examples of that in a minute. And finally, the good thing about natural products is if they come from microbes, you can easily scale them up. And if you know the genetics, you can often modify them as well. I'll give you examples of that later in the presentation. So we're interested in biodiscovery. And my favorite place for collecting things is some beautiful parts of the Atacama Desert in Chile. So I've been working with Juan Asenjo and Barbara Andrews there for some time. We look at that for bioresources. So in this case, interesting microbes, or deep sea, we might get interesting sponges. And we would look at the chemistry of the organism directly, but also quite often the DNA and the RNA sequence data can be looked at to see what the genome contains and what we may not be seeing in terms of chemistry. And again, that's really exciting when we can express those molecules under certain conditions. We do some kind of screening to test for activity, and hopefully in the future, you'll be able to make a product out of that. And we've never made it to the clinic yet, but we are working on compounds that are exciting and often handed on to a synthetic chemistry group to do the next stage. So we're interested in all these environments. We started off particularly in coral reef environments, but moved on to deep sea, cold oceans, and desert environments by working with two very famous microbiologists. One is called Mike Goodfellow. Newcastle is a world famous taxonomist. Another one called Alan Bull from the University of Kent, who is a very well-known microbial cultivation expert. And we're in the group, we're pretty open to what we look at. So we still look at some marine invertebrates. I mentioned nudibranchs. My colleague Rana Abel is an expert on fungi, both edible and non-edible. So he likes to look at fungi from the sea and from the desert. I like the bacteria, and my colleague Wael Hussain likes cyanobacteria after we worked on this together for maybe 10 years. And we see lots of new chemistry arising all the time. New structural families, new biological activity. It's a lot of fun to see the kind of diversity that we can get out of marine and desert organisms. So here's two examples that have gone quite a long way. This is from the pharmacy project that Giselle mentioned already. And the first compounds are from a marine fungus. And these two compounds were tested in zebrafish larvae to see whether they were able to prevent seizures caused by the treatment of the zebrafish. So again, they're a model for epilepsy. That was then taken immediately because the molecules were shown to be active in the zebrafish model, very effective. They were bioavailable and non-toxic. So that's what you can get out of zebrafish information. We're able to test it in animal model very quickly. And that was a very potent inhibition of seizures at a very low dose level. And they both showed excellent ADMET profiles again. So this is important that they were drug-like, basically. We wrote a paper, we filed a patent. The next stage is to make enough. So the US National Institute for Neurological Diseases has asked us to provide them with 500 milligrams of this material for the next stage of testing. Unfortunately, that's very tough. It took us a year to get 35 milligrams from the fungus. So what we've done now is we've teamed up with Jakub Svenda in Prague University in the Czech Republic to make this molecule. So it's taken him a couple of years, but he's getting there. So again, he's a world expert at this kind of synthesis. So it's nice to see this going to the next level. These molecules here were from sponges. Okay, so they were already in our compound library, and we sent them over to Luis Betana's lab in Spain. And we had this potent anti-Alzheimer activity and this anti-inflammatory and immunosuppressive activity in the same molecules. What happened at that point is that Dan Romo, who is a very good synthetic chemist from Baylor University in Texas, was able to synthesize groups of molecules which separated that activity. So now we have a series that is anti-inflammatory and a series that's anti-Alzheimer's disease, which is pretty cool. They have taken this very seriously, and they're scaling up the synthesis in Texas, and I periodically get an update as to what's happening with that. So I'm very excited by that. And again, it was published in a good journal, and the patent was filed shortly afterwards. Again, it's very exciting to see this go to the next level. And again, I feel good that one of my lead molecules has gone into the next stage of development. So I'd like to talk today particularly about a group of metabolites or specialized metabolites called ribosomally synthesized and post-translationally modified peptide natural products, which are my favorite group of molecules. So what happens with these? You have a small gene cluster, typically something like 10 kilobase pairs, which is much smaller than NRPS or polyketides or isoprenoid metabolites. And it's composed of several genes which process the precursor peptide. The precursor peptide is made of a leader sequence, some kind of signal, a core peptide which is modified, and another signal. And this can vary. Sometimes the leader is at the end, so it's not a leader, it's a follower. Sometimes the signals are not there, but the core peptide is always the bit that gets modified. So in this case, you get some post-translational modification by the enzymes that recognize this precursor peptide, modify something, and often you get this cleavage from the leader peptide, you get this maturation, as it's called, and protease, and you get a mature folded active peptide, which is really the final product that we see in nature. So there's lots of them, and I don't know how well this is coming across, but these molecules here are very exciting. So for instance, we have things like the telomestatin, which has got 7 heterocycles in a ring, or is it 8? 8 heterocycles in a ring. We have this thing called the sapomycins, which are long linear molecules with some modifications. The world's largest ribosomal peptide is called proteusin, which was found by Johan Peel. Food preservatives, LASU peptides, thiopeptides, which can be good antibiotics, and the ones that we're going to be talking about today mainly are the cyanobactins. I'll use that as an example, but in order to lead to that, I'm going to start off with some information or some studies we did on LASU peptides. So let's go to the driest place on Earth. Again, I was fortunate enough to visit Chile 3 times now, and going to the desert, I'm sort of indicate roughly where we went, sort of this area here of the desert, and we went into this top corner here where 3 countries meet, and it's about 5,000 meters in altitude with hot lakes and everything else up there. So it's a beautiful place to go. Most important thing you need to know is that it's very dry there, and what it says is an absolute desert. That means there's been no rainfall for about 1 million years. It's been dry for about 23 million years. And, you know, the dryness is defined by the fact that you get more evaporation than rainfall by a factor of 500. And the reason for that is simple. On the one side, you have this coastal mountain range here, and fog will rise up to the top of the mountains, but not travel across the mountains. On the other side, you have rainfall from the Andes Mountains, and it meets a geological fault underground, and that prevents any water from getting to the central depression. But the central depression is the driest place on Earth. It's really incredible place to visit. There really is no water there. And when it does rain every once in a while, you get amazing flowers and everything else arising. So here's a view from 4,000 meters down, looking down upon the Salar here, the Salt Lake. The cacti are twice my height, so about 4 meters. Quite an amazing view. And I work with these people here. So this is Mike, this is Alan, and this is me. And it was Juana Senjo and Barbara Andrews who are our hosts for that visit. But I think it's a really beautiful place to visit. And we're really lucky to find so many interesting microbes there. So this is one of the strains that we found. It's called Streptomyces leeuwenhoekii C58. And again, it was interesting. We did do the genome of a related strain and found that it had, I think, 30 or more gene clusters, of which one of these was this Atacama strain, this strange peptide, which had 15 to 16 amino acid residues. So first of all, it's a large molecule. So we managed to get a very good accurate mass. It's always good to get this, you know, and it tells you from the formula already that you probably have a peptide because the N and O count is roughly the same. And it's roughly the right number of carbons and hydrogens for an amino acid. So it's just about probably 16, 17, 18 amino acids, whatever. And this is a beautiful, I love showing NMR spectra when they're good. This 700 megahertz cryoprobe, so we had to use somebody else's machine, but you can see the beauty of the spectrum is that it's highly resolved and well dispersed. So we have really good from 0.5 to 9.7 roughly ppm, and there's good separation between the peaks. Again, normally we see, in this case, we see 15 NH groups, see the phenyl groups, lots of alpha protons, lots of beta protons, and then the rest of the methyl group. So it's quite complicated looking molecule. But when you do the structure, do the assignments in 3 dimensions, you can do the structure as well. So this is done together with another group in our medical school who do this kind of 3-dimensional modeling. And what you can see is it's a molecule that contains a loop, a connector, and a string. And the string goes through the center of the loop. So it's a Lasso peptide. We don't know what their activity is. They don't typically have very good antimicrobial activity, but they do have interesting structures. And again, if you think of why does the string not pull out of the loop? The reason is that if you look at it from a different viewpoint, the residues that go through the loop are incredibly large, and that the string really can't pull back through the loop without any problem. So again, it's an interesting type of molecule. It also asks questions about the biosynthesis. So it's very complicated to get that molecule synthesized without having the string through the loop before the loop closes. So it's a very exciting biosynthesis, and that's been worked on recently. So we did it the wrong way around. So we did the NMR spectrum analysis and figured out the structure, and then realized that the genome would have got us there much quicker. Again, you can see this is related to another molecule. You can see the relationship here. They're very closely related, 15 residue Lasso peptides. So the easier way would have gone the other way. We would have gone the bacterium, get the genome, look at the gene cluster, predict the product, right? That would have been the simple way to do it. But this was 5 years ago. And now we did exactly the same thing in less than a year. We got this from the beautiful lake, Sala de Huasco. We got the genome, we got the gene cluster, we predicted the structure, and we confirmed it in 3 dimensions. Again, turns out this was unique because it was the smallest loop containing compound that we had. Okay, and then again, Scott may have shown you this as well if he's talking about SMS to you. But anyway, it's nice work that he did to show that actually normal fragmentation of these peptides just allows you to cleave off the chain, but not break up the loop. And that makes it, you see nothing beyond this point. You see 746 and then 701, and that's it. There's nothing else you can see, which is difficult if you want to look further down in the loop to confirm the structure. So what you do instead is this HCD methodology. I think it's high energy collisional dissociation. Trying to say that. And what you get with that is it breaks it up in certain ways. Sometimes you get a T-shaped molecule, but sometimes, if you're lucky, you get a linear molecule, which allows you to then fragment that and get the full sequence of the compound in the mass spectrometer. That makes it much more easy to determine whether or not your biosynthesis is producing the compound you expect. And again, you get the entire sequence. You can count all the way along as you go down, right? So it's nicely labeled here by Scott. And again, the microcycle opening, you saw 33 out of the 35 ions we expected. Again, you can break this in any position, so 1, 2, 3, 4, etc. And each one of those will give you the whole peptide, but also then fragment into smaller units. And again, it's really a powerful set of methodologies to determine the structure of these complex peptides. So that's enough about the biosynthesis and how we can analyze the structures. I want to talk now a little bit about how and why we're interested in the kind of molecules in our company and where the background came from. So the reason for looking at large molecules is because we're looking at drug discovery from a different viewpoint now. So normally we're used to looking at small molecule drugs that fit compact binding sites. So things like ion channels, kinases, etc. And there's lock and key classical drug interactions, right? So there may be about 500 to 700 targets in the human body that are susceptible to this kind of treatment. On the other side, where there's extended binding sites, and normally protein-protein interactions and class B GPCRs are affected only by native proteins and antibodies. So we see a lot of drugs right now that are antibody based, but they're sometimes, difficult to administer, and they're quite expensive. So the world's biggest selling drugs are in fact things like Humira, which is an antibody for arthritis, inflammatory disease, about $10 billion per year. On the middle part, we have this wonderful class of molecules which we call natural products and macrocycles or cyclic peptides. And they have the properties of being able to interact with these very large targets, but they also have the properties of small molecules in that they can be administered in the same way as small molecules, as orally, as drugs, and they can affect, they have a different effect compared to small molecule drugs, so they're quite cool. And the advantages, we use cyclic peptides because they can modulate these challenging targets, they can cross membranes, they're stable to metabolism quite often, and they have very good target affinity and specificity because they already have this entropic advantage by being in the right conformation. But synthesis can be challenging. So let's see how we can address that. So protein-protein interaction, you would have to think about a very simple model that I've drawn here. You take your macrocycle, it meets with one side of your protein-protein interaction and blocks the interaction, preventing the disease state. The types of diseases that we're thinking of, anything from inflammation, cancer, epilepsy, hypertension, Alzheimer's disease, etc. So it's a target that is relevant to many, many different types of diseases. So we started off with a marine organism, and the organism is this organism sea squirt called Lysicline patella, which is an ascidian or a sea squirt, and it contains a blue-green alga called prochloron, which we now know is responsible for the formation of the patellamides, these small molecules here that you see, or these cyclic peptides that you see here. So this is interesting because these molecules were originally of interest to us because they were able to reverse multiply drug-resistant cancer cells. So if you added a small amount of this molecule to resistant cancer cells, they would reduce the resistance by blocking the protein-glycoprotein drug efflux pump. So that's interesting. So again, we thought that was a good reason for making these molecules. And we did a long, this whole project took between 1996 and now, so it's a very long-term project. But in 2004, we cloned the biosynthetic pathway, which is what I'm going to tell you about now. But the key is that these molecules are very complex, and we know that they're ribosomal peptides, and they have a number of modifications. One of them is, for instance, a heterocycle formation or an oxidized heterocycle, epimerized stereocenters, and macrocycles, or prenylation. So you have a lot of alternative ways of modifying a molecule in a very short biosynthesis. So our pathway, so we published in the same year, there's a long story about this, which I don't want to tell you right now. So what you have here then is this first enzyme is the leader or the precursor peptide called PAT-E, and it contains this cassette or core peptide, which is modified first of all by the heterocyclase, which is the enzyme that makes these into heterocycles. I'll explain more of that in a minute. That then is followed by cleavage at this end, followed by cleavage at this end, followed by macrocyclization. And then we don't know if this epimerization is spontaneous or not, but we think it is, followed by oxidation to make these things into thiazoles rather than thiazolines. So the enzymes that we have, first of all, I worked with a very talented crystallographer called Jim Naismith at St Andrews University and now at Oxford University. And what we see is the enzymes are able to do different things. So the first one is called TrueD. It's able to take only the cysteines into thiazolines. On the other side, we have PAT-D, which can take the cysteines, threonines, and serines into oxazolines and thiazolines. And the enzyme itself has a very novel fold, as you can see here. It contains a domain for binding ATP, a domain for binding the protein, a peptide clamp, and something that binds to the leader sequence. Again, so it has 3 different domains doing 3 different jobs. It's the first enzyme of its kind to be made of a single single protein. Normally, these are found in 2 or 3 bits separately that bind together. The next enzyme cleaves at this side of the molecule here on the left-hand side. And this was the first enzyme we in fact crystallized and identified. And it's not particularly efficient enzyme. So We did use it for a bit, but it's not a great enzyme, but we know how it works. The next stage is the macrocyclase, which is an exciting enzyme, which essentially takes off the right-hand signal and turns it into the final macrocycle molecule after some kind of oxidation and epimerization taking place. So the way that this works is very exciting. It looks like a normal protease. Okay, so we have this extra bit. So normal protease is given as 1 dBi here. The insertion loop is about 35 amino acids, and it does an amazing job. So what it does is it protects the normal active site. The normal active site is made of histidine, serine, cysteine. Sorry, histidine, aspartic acid, serine, and the cysteine is doing something else. And it protects that. It hangs over the top of that active site, as you can see, which is quite unusual. So what we saw close up is that what you have is this, the AYD in the leader peptide, on the signal sequence, sorry, binds very tightly to the leader, to the lid, or this extension of the amino acid sequence in the protein, the enzyme, and it allows only folded substrate to bind. So once this folded substrate is bound, it cleaves it just here, and allows the N-terminus to fold back on itself here, forming the final product. Again, so that's a really neat, simple mechanism. So literally it causes hydrolysis, but by keeping water out of the way, allows it to cyclize onto itself. So it's the N-terminal will bind onto the C-terminal that's been cleaved and freed up by this enzyme. So it's a really simple but elegant mechanism. So what do we do first of all? We were able to modify this this process by taking something very interesting. So we took the leader sequence, the start, the core, the stop sequence, and we decided that this first enzyme was very poor. So we put in a protease signal so we could do a faster cleavage, and we put in a his tag so we were able to isolate the molecules really quickly. So first of all, we took a core sequence, we added the heterocyclase enzyme, we modified the serine, threonine, and cysteines to make the heterocycles, and we could purify the compound immediately using our his tag, using an IMAC column. The next step, we used the first protease, so not the PAT-A protease, but just ordinary trypsin to cleave at this point. And then we're able to purify again using the his tag, and then the final cleavage takes place, the macrocyclization gives us then the final product that we looked at here. And the beauty of this methodology is that if you look at it, look at the isolated compound from nature, you look at the purified compound from the biosynthesis, the purified compound from the biosynthesis is cleaner than a natural product. So it's really, really good. We are able to determine a 2D NMR of this molecule and show that it's identical to the natural product in every respect, including its chirality, using circular dichroism. The other thing that you could see is that the enzymatic oxidation to the final product is really efficient with this oxidase enzyme. That's a really neat step, I think. So again, this could be a great chemical reagent for people who need to do this step. Normally in synthetic fields, it's just very slow and very low yielding, about 30%. Oh, it is 100% in 2 hours. Finally, some ideas that we had. We had another series of similar compounds. And here, what we're doing is we're adding a prenyl group and reducing the conformational freedom. So what's happened here is these two tryptophans are modified to include an extra ring and this prenyl group. Again, it makes it a very, very interesting molecule altogether. So in principle, what you can do now is you can take a single substrate that you have. So you make this precursor peptide with a modified core sequence. You can take a different enzymes that do heterocyclization, some that only do cysteine, some that do cysteine, serine, and threonine. You make your compound, you cleave them off, and then you can do macrocyclization, oxidation, either just oxidizing the thiazolines to thiazoles or the oxazolines to oxazoles as well. You get that over here. Or you could just do one side, do your oxidation and do your prenylation. So you can have lots of different molecules from the same starting material. So I've shown 6 here. I think there are 7 or 8 we can make using this methodology. But the first thing we did is to publish a paper on this, showing how diverse the range was. So 6, 7, 8, and 9 amino acid macrocycles with 2 to 4 heterocycles, using 11 of the 20 natural amino acids, all different types. So again, it's a very cool pathway. It was published some years ago, but again, still very powerful methodology to apply. But then we discovered a really neat addition to this whole thing. When we did the crystal structure of the heterocyclase together with the leader sequence, what you see is the leader sequence binds like this. And that's needed to activate this enzyme to be to work. So what we did then is we decided we play a trick played by my friend Wilfred van der Donk in Illinois, which is to tie the leader sequence to the enzyme itself. And now the enzyme becomes a chemical substrate. It becomes more chemically stable. We know that. Easier to express, and it's permanently activated. So again, what we need to do now is feed it the substrate enzyme, the substrate peptide, and we need to feed it the ACP to make it work. So what you do then is you take your synthetic peptide of the bit you want to the core peptide and a 3 amino acid signal sequence, okay? You use your engineered heterocyclase I just mentioned, you get your heterocycles formation, you use your engineered macrocyclase and the new oxidase as well, you get your final product. So again, you can use this very simply to go to the next stage. So let's see, we managed to take the process into a one-step, a one-pot procedure as well. So what you do is you get your synthetic substrate with even unnatural amino acids in. We use our enzyme, our engineered enzyme to make the heterocycle. We use your engineered heterocyclase, and we get our final product here, which we can modify still at the N3 position. So the azide can be reacted using click chemistry. So in this case, we went back to our roots and we said, well, actually, the reason we wanted to do this was to test it against PGP. So we made a family of molecules against PGP and we tested it. Unfortunately, the natural product is as good, if not better, than our best compound. So again, nature already had the right answer, but what it showed is that we can modify these, we can make these molecules, we can make them bioavailable, and we can modify the activity using chemistry. So what we see here is that we made the activity significantly worse rather than better, but the principle is still the structure-activity relationship, although this is not normally the way you want it. So we could use that azide group to attach things to. So again, this is the molecule I showed you a minute ago, but no heterocycle at the end. And we use that to introduce a fluorescent molecule inside a cell. What we can see now is you can do click chemistry on this, attach a fluorescent linker that then goes into the cell here. You can see it fluorescing inside the cell. So the other thing that we tried then, this is Emilia's work. And Emilia's decided to take some different non-amino acid substrates. So first of all, just N-methyl. Then this group here, which contains a carboxylic acid and an amino at the other end. So again, that still works. Okay. Then a sugar, then some long fatty chains, then some longer oxygenated chains like this. And in each case, you can see that the molecule you can make it in fairly high quantity and it's quite diverse. So again, the point is that we don't need to have all amino acids to make this process work. So at this stage, we started to the idea of creating a company. And in 2015 and 16, we had money from Scottish Enterprise, which is the enterprise arm of our Scottish government, called the High Growth Spin-Out Program, to try and develop this to the next stage. And we created a company called Riptide, which I think I told Giselle about the last time we met. And Riptide was doing quite well. We had, you know, the two scientists, inventors. We had Bill Primrose, our CEO, and we had Andy Bent and Andy McEwen, who were our scientists in the lab. We even won the Scottish Life Sciences Innovation Award in 2015, which is great. You know, you have a new idea, you have a company that seems to be doing well. Unfortunately, they hate it when the company doesn't succeed and they award a company that doesn't do well. Of course, we had a problem with industry deals. So industry was all being swept up by a big Japanese company called Peptidream. But what happened subsequently is that those companies came back to us to say, actually, your idea was really quite good. We'd like to work with you as well. And then the company was gone. So we had to start again. So my colleague, Jim Naismith, had moved to Oxford at this point, and we decided to place the company in Oxford because of the very good entrepreneurial environment and the good biotech environment that's around Oxford. So we hired our first scientist, Arode Anijurengo, and now we have Paddy Killoran and Wouter van der Linden joining us as well. So again, we started a couple of years ago, but really only getting going now. And we're able to get to the labs enough time to hire a CSO who will join us next week. There's Arode. She's planting a tree on behalf of the company. That was what Oxford does right now. They put a tree and they put underneath it to the company that planted that tree. So the technology is really simple. It just relies on the ideas I mentioned before. We have the technology, the substrates. So you make a peptidic containing substrate. It hopefully will appear. Oh, something disappeared. Okay, so you get your modified core peptide. You get your heterocyclase. Yeah, you get your macrocyclization, oxidation, perhaps prenylation, and further chemistry. So again, we have the best of both worlds. We have both the best of the synthetic chemistry and the best of natural product chemistry and enzymic reactions. But the key is that diversification. You take the same substrate, you take different cocktails of enzymes with chemistry, and you can make a whole range of different products that are closely related but different. So you can explore structure-activity relationships very quickly. I'm going to stop with that, but thank the large number of people who have been involved in this. So for the chaxapeptin, the LASU peptide, I want to thank particularly Sumaya and Mustafa, who worked on the structure before the genome was known. And for the Huasco peptide, particularly Carlos and Scott worked on that structure. And for the cyclic peptide stuff, I really want to thank Wael, who's been my colleague for I don't know how many years now. He was my PhD student, graduated 2006, and has been working here ever since, and has got himself a £1.3 million grant recently to further his own research. So he's brilliant to work with him. Unfortunately, our first scientist at the company, Andy Bent, died of a brain tumor, sadly, a couple of years ago. So it's very sad to lose him. But now, the new company, we have Bill, Jim, Arode, Paddy, and now also Wouter. Of course, this couldn't be done without money from lots of different people. I just point out, European Research Council was a grant to myself and Jim Naismith. We've had European funding, as I mentioned already, but the most incredible funder always for me is the Leverhulme Trust. Leverhulme Trust is a research funder that funds only high-risk research, and I've had funding from them twice, and it's always been the most exciting research that I've done, including this one here. So thank you very much for listening, and if you have any questions, I look forward to your questions. Thank you.
Thank you, Marcel. I would like to open the opportunity to my colleagues to raise their hands and make make their questions. Until I have some questions from the public, I would like to know, Marcel, how difficult it is to work in Chile in terms of accessing biological material.
Okay, so I mean, because we work with the laboratory at the University of Chile, we've always worked through them and with them, and every single paper we have with them is in collaboration. So they don't have what you call it again, any Nagoya Protocol ratified in Chile, as far as I know, but we always have agreements between our institutions that are Nagoya compliant. Mm-hmm. So it's really important to recognize that fact. But the times that we have worked on materials from their lab, it's either been scientists who've come to visit us and work with us, or we've gone to them, or the work has been co-published at all times. So it's really quite fun to do that.
Mm-hmm. The other question that I have is, being that, for example, in the Atacama is nearby the salines, and do you happen to see compounds higher in halogen content as opposed to that might be very interesting to evaluate.
Yeah, so I mean, the Salar, we were expecting to see more chlorinated compounds. We didn't see many of them. So there was one experiment that we never completed, and so the difficulty has been that we can see a chlorinated organometabolite using another technique called ICP-MS, mm-hmm. and we see a spike. But unfortunately, it's expressed in such small quantities that we can't see it by normal MS, so we can't isolate the compound. So it's a challenge. So we never resolved that. We do see a chlorinase genes in the one of the streptomyces from the Atacama Desert. So it's definitely there, but it's going to take some time to tease out why it's not producing it when we want it to.
I was looking into a graph, and I don't know if you're familiar with the Earth Microbiome Project from Robert Knight. and they're making a graph on halogen content of the different actually natural products that, well, metabolites that they are seeing, and that's I mean, it was fantastic to see how different halogens could be, you know, involved into this. And I was keen to see, I mean, they're real nearby, the photo that you took into to the solar, and I don't know if there might be halophiles, kind of microorganisms that you may be seeing.
Yep. I would assume that they're there, but we haven't found them yet. Definitely.
Okay. I have the first hand here, Daniel.
Hey.
First of all, thank you very much for all the for the talk and very impressive work. I think you mentioned that you synthesized some substrate for the elicitation of biosynthetic gene clusters. What kind of substrates do you use for elicitation of that kind of clusters?
So we're really interested in, at the end, the heterocycle that you get on the C-terminus is, it would be nice to replace that with something different. So we know proline works, we know that thiazolene works, oxazolene works, etc. So we tried some other ideas, like, we published this already. We tried some 6-membered heterocycle. It doesn't work so well, or N-methyl, it doesn't work so well. We tried to use what's called a pseudoproline. So it's like a serine that's been modified to have a dimethyl carbon compound. And that, in principle, does the cyclization, but we can't, no, it doesn't do the cyclization. We can make it, and then we can't do anything with it. So we remove the 2 methyl groups, and now it cyclizes nicely, but now we can't remove the methyl group. We can't remove the protecting group. So yeah, it's a lot of interesting things. So we published some of these papers in ChemCom 2-3 years ago, I think. So, but if you want, I can send them on to you if you're interested in that kind of stuff.
Okay.
And I have other question just about Streptomyces C58.
Yeah.
When you first grew it, did it express the chaxapeptin, or did you have to do some kind of elicitation to make it happen?
So this one was quite straightforward. We saw it right away, but there was a related strain called C34, which had this. When we looked at the genome, it should produce a similar or the same LASU peptide, but it didn't. So we had to play with the conditions. Quite nutrient limiting conditions seem to be allowing it to produce, but we've had other ones where these ribosomal peptides don't get produced until you take the pH down to pH 4 or 5. You increased another one. We had to increase the temperature quite a lot. So
it's like
heat
an
shock.
osmotic approach.
Yeah, different conditions. So I mean, the funny story is the one that where we heated it up was an accident. So my colleague Wael went on holiday and he left a cyanobacterial culture at 18 degrees, which is the optimal for that particular strain. And he came back and somebody turned the room to 30 degrees. And the organism had died and gone white and produced the compounds. So what we did then is we had to reproduce the same conditions. All we did is really, he said it worked in one day. So literally he took the temperature from 18 degrees to 30 degrees for one day and he got the same result. But that's just lucky.
Thank you.
Thank you, Daniel. Alice?
Hello to everyone, and thank you for your talk. I just have a curiosity, since you have all this expertise in peptides, and what you thought about the compound of PharmaMar, plitidepsin, and the idea that maybe some of your compound, and you have families, do some dockings or some experiments or something related to SARS-CoV-2 that is now the hot topic. But related, having you cyclopeptides and the work of PharmaMar, I'm just wondering if you thought about this issue too.
Yeah, so my company's been discussing that particular compound to look at. We did actually, what we did do was to design a family of compounds against COVID March last year. And we made 100 compounds, and none of them were active. So yes, we tried. We're not stopping there, but we did. The problem is with structure-based drug design, it's really hard to do without a starting point. But now with plitidepsin, we have a starting point, so we can start to think about molecules that are like that, that we can make that. And so the specialization of our company is that we can restrict the conformation of the molecules so well that it always is a hairpin or is always in a funny shape. So that's the key to what we can do. So it will change the well, it'll give a much greater entropic advantage because it can bind more readily, and it will. We've shown already that both the polar surface area and the log P can be controlled using the technology. So it's really, that's the key. So we have these three angles. We have conformation, log P, PSA, which is exactly what we want. So yeah, we keep trying for the next pandemic, for COVID 2023.
Thank you, Ali.
Yeah.
Adriana?
Yeah, so I think Giselle has my paper on COVID antiviral discovery. I think I sent it via LinkedIn, right? You commented on that, so.
Thank you. Adriana?
Hi, good morning. Can you hear me?
Yes.
Yes.
Awesome. Thank you so much. Nice to meet you, Marcel. And I'm glad that you like Costa Rica when you can visit. So it's nice to hear that. So I have a really specific question. I don't know if you can give me an advice or not. That'll be awesome. I just have a question. If you can let me know if there is a specific database that maybe we can use to research natural products for drug discovery. Is there like one database that you will recommend? Because I've been going through several databases and there is just one that has this subset about NPs, but the other ones are maybe a little bit difficult to find. So I don't know if you have any advice on that topic.
Yeah, I mean, we use all kinds of things. I mean, there is the Dictionary of Natural Products, which is really expensive, and it's not comprehensive and doesn't do stereochemistry very well. We use NP Atlas quite a lot, which is Roger Lillington's database. Okay, so it's just called Natural Product Atlas. It's quite good. There is one in Sweden called ChemGPS, right? So just like a global positioning system. And that's, I'm not sure if that's still running. So a lot of these databases are very short life. ChemSpider has some subset. It's not great.
That's the one basically that I found the best, but yeah.
That's what we tend to use. And we use also Reaxys, which is a pay-for database, but the UK signs up to that as a consortium agreement, so everybody in the country gets access to that if they're in a scientific institution. But there isn't really a good solution, I have to say. So we tend to do a lot of searching in different databases to find things. I think if people start to contribute to NP Atlas, it may be a solution to a lot of things. But again, it needs to be comprehensive. So maybe there are 150, 160,000 natural products out there. And there aren't really any good solutions. So MarineLit was good for marine compounds, and there was Antibase for natural products of microbial origin, and they combined for a while, but now one of them is commercially available only through the Royal Society of Chemistry, and the other one is I'm not sure if Antibase is still going. So yeah, I'm sorry, there's nothing good I can say at the moment. It is difficult.
Well, the thing is that now I know that it wasn't just me struggling. It's just how it is.
Yeah, we should improve it. I mean, we have a big discussion in our country right now about getting together all the natural product people. I spoke to other people in Switzerland recently, and they said basically we need to improve this as a network, international network of natural product scientists to get these databases done. We should be able to do it for free, and it should be like the biologists or the open access databases and stuff.
Awesome. Thank you so much.
Thank you.
Is there any other question?
Thank you for the link. Yeah.
Okay. But I do have a question related to Danielle's question actually, and it is when you moved from the classical, I got the natural product and then you look for the biosynthetic pathway, and now you look for the biosynthetic pathway, and then after you go to look for the natural products. How many conditions do you test before you actually isolate the compound or at least detect it?
Yeah, so about 8 or 10 normally. as a minimum. So it's quite a few. We've actually had a student, he's did a really good job recently. What he did is prepare a 96 well plate with 12 different conditions and 8 different strains. And he just did that, and then he does quick LCMS on pooled samples, and he gets good results out of that one. So yeah.
Well, when you have Michael Goodfellow on board,
yeah,
I think you have the whole recipes.
Yeah, we have the recipes. No, I mean the good thing about that is that often he sends us strains that are already pre-curated, so he knows they're really good. And what we do quite often is we take from him maybe 25 strains, and we do a quick run through of all the different conditions. And we see that quite a few of them are maybe closely related, or they don't give any good chemistry. And then we've got the optimal conditions, and a student will work maybe on 5 strains for their entire thesis and maybe get 50 compounds out. But out of those, maybe 10 are new. So it's actually a pretty good hit rate compared to other methodisms. So my approach is always work with the best taxonomist you can find, and that works well.
Yeah.
But he's retired now, really. So yeah, I work a lot with his students are going all over the world, and they often contact me through Mike, and then we get some interesting chemistry.
Yeah, are you going to move towards synthetic biology, as taking genes from one part? And
yeah, we tried with this pathway with a company called Ingenta, which is a synthetic biology company, to develop a framework for making these molecules quickly using a single strain of organism. And it works 3/4 of the way, it works, and then it breaks down. And the reason is that the last enzyme is so rubbish, it's so slow, that the organism dies before the whole thing is done. So yeah, it can be done, but it's really tricky to get it right. So it's not quick. It took us 3 years with 6 people working on it to not make it work. So yeah, synthetic biology is great, but it's hard work.
Mm-hmm. No, that's okay. What about the non-ribosomal peptides, Marcel? You are living a whole world outside your.
Yeah, so I like the ribosomal peptides just because they're so simple to understand. I can look at the substrate, I can look at the product, and I can understand the chemistry that happened. With non-ribosomal peptides, it's so complicated, and there's so many people fighting in that field that we got into this field at the very beginning, right? So it was very hot field. When we got the first crystal structures in 2010, we were ahead of the field. So that was great fun. So now we're still working together on these things, but a lot of other people have overtaken us because they have bigger groups. But yeah, it's nice to be the sort of the foundation of a particular field, and then just enjoy that. But NRPS and PKS. So I mean, my colleague Haideng is good at NRPS, but mainly PKS and things like that. So, but we seem to be all focusing on RIP right now. So even the European project that just got funded will be RIP focused because we have Jörn Peel and Manix Medema on board.
Okay. I don't know if there is another question. If not, then thanks again, Marcel, for being so kind to give us this talk.
Thank you, Giselle.
And try to be in contact always.
Yeah. I hope maybe we can travel again in the next year or so, and then we should see each other in real life.
Okay. Thank you very much. And
thank you, Giselle.
Thank you all for attending this talk. Thank you very much. Bye-bye.
Bye. Thank you.